Welcome to the deep dive. Today we are diving headfirst into one of the most magnificent, precise, and frankly dazzling processes in all of biology.
We're talking about meiosis and the sexual life cycle.
Exactly. And you see the results of this everywhere. Look around. You, your family,
Hmm.
offspring always inherit traits from their parents, but they're never, exact copies.
Never clones. And that tension, that space between resemblance and variation is really the engine of evolution.
Our mission today is to give you the ultimate shortcut to understanding this entire biological chapter. We want to go from the tiniest gene all the way up to how entire species stay stable.
Right. We're translating all those complex diagrams and formulas into something you can actually visualize just by listening.
So what's the core idea, the one thing to hold on to?
Well, it's this. Meiosis is a special kind of cell division. It's a reduction division. It has to cut the chromosome number in half.
It has to. Why?
Because it needs to perfectly counterbalance fertilization, which is the process that doubles the chromosome count. It's a beautiful balancing act that keeps a species genetically constant over generations.
Okay, so let's start with the blue point of heredity. Where is all this information actually stored?
It's all packaged into units we call genes. They're written in the language of DNA, and they program basically every trait we have.
And these genes aren't just floating around randomly, right? They're organized.
Highly organized. Each gene has a very specific address on a chromosome. We call that address its locus.
It's locus. Okay, so when we talk about the cells in our bodies, we need to make a key distinction.
We do. First, you have what are called somatic cells. These are basically all your body cells, skin, muscle, brain, everything except the reproductive cells.
And for humans, these somatic cells have 46 chromosomes.
Correct. Then you have the specialized delivery vehicles, the gametes.
Sperm and eggs.
Sperm and eggs. They're the only cells that can pass genes to the next generation. Meiosis is all about making them.
And how those genes get passed on. That defines the type of reproduction. You've got asexual reproduction, for one.
Right, where a single parent just copies all of its genes. Think of the yeast cell budding, or a hydra.
And the offspring are.
They're clones, genetically identical. The only way you get any difference there is from a random mutation, a copying error.
But then you have sexual reproduction, two parents, and here, the whole game changes.
The whole game. The goal is no longer perfect copies. The goal is unique combinations of genes. It's all about variation.
Okay, so to really get why that variation is so powerful, we need to look at how the chromosomes themselves are organized.
Absolutely. So if you were to take a human somatic cell, one of those with 46 chromosomes, and you arrange all of them in order by size and shape, you'd get this visual map.
That's the karyotype.
That's the karyotype. And what you'd see immediately is that they come in matching pairs.
We call these homologous chromosomes or homomogs.
Exactly. You get one chromosome of each pair from your mother and the other from your father. They have the same length, the same shape, and they carry genes for the same traits at the same loci.
So they're not identical, but they're the same type.
Perfect way to put it. Now, there is one major exception to this perfect pairing.
The sex chromosomes.
The X and Y. Females typically have two X chromosomes, which are homologous. Males have one X and one Y, which are not. The other 22 pairs are called autosomes.
This brings us to some really critical terminology, diploid and haploid.
The foundation of all this. Since your somatic cells have two full sets of chromosomes, one maternal, one paternal, we describe them as diploid or 2N.
So for humans, 2N equals 46.
Right. But the gametes, the sperm and eggs, they only have one single set of chromosomes. They're haploid or N.
And for humans, N is 23.
And here's a crucial rule to remember. Only diploid cells can go through meiosis. You can't have something that's already been halved.
Okay, let's trace this through the human life cycle. It starts with fertilization.
Right. A haploid sperm, with N23, fuses with a haploid egg, also N23.
Which creates the very first cell, the zygote.
And that zygote is now diploid. It's 2N46. It then divides over and over again through mitosis to build a whole multicellular adult.
But to keep the cycle going, we can't just keep doubling the chromosome number.
Exactly. So in the adult's reproductive organs, the testes or ovaries-specialized germ cells undergo meiosis.
And that's the reduction step. It takes the chromosome sets from 2 back down to 1, creating new haploid gametes.
And the cycle is ready to begin again. Now, this basic alternation is universal for sexual reproduction, but different life forms play with the timing.
For us animals, the adult is diploid and only the gametes are haploid. Simple enough.
But then you look at plants, they do something amazing called alternation of generations. They have both a multicellular ziploid and a multicellular haploid stage.
Wait, a haploid adult, how does that work?
The diploid plant, the sporophyte, undergoes meiosis to make haploid spores. These spores don't fuse. Instead, they grow by mitosis into a whole new haploid plant, the gametophyte.
And that haploid plant is what makes the gametes.
You got it. And then at the other extreme, you have most fungi. For them, the only diploid cell is the zygote,
So it's the total opposite of
a
us.
complete reversal. That zygote immediately does meiosis, and the rest of the organism's life is spent as a haploid adult.
Hmm.
But the core principle alternating between N and 2 always holds.
All right, let's get into the mechanics, the real choreography of meiosis. How does the cell actually pull this off?
Well, it starts out looking a lot like mitosis. You have interphase, and during the S phase, all the chromosomes get duplicated.
So we start with one diploid cell, but with 46 duplicated chromosomes.
Exactly. And then it goes through two divisions, back to back, meiosis the third and meiosis the second.
And meiosis the third is the really unique one, the reduction division.
That's where all the special events happen. It's designed to separate the homologous pairs.
Okay, step one is prophase I.
The busiest stage by far. The chromosomes condense, but then something incredible happens. The homologous chromosomes, the maternal and paternal versions of chromosome 1, for example, find each other and pair up precisely, gene for gene. This is called synapsis.
How do they stay together?
A protein scaffold called the synaptonemal complex forms between them. You can think of it like a molecular zipper, holding them tightly. And while they're zipped up, they do something called crossing over.
This is where they actually swap pieces of DNA, right?
Literally. A piece of the maternal chromatid breaks off and reattaches to the paternal chromatid, and vice versa. When the zipper dissolves, you can see these X-shaped connection points called chiasmata.
And those chiasmata, they physically hold the pair together for the next stage.
They do. Which brings us to metaphase there. The homologous pairs line up at the center of the cell at the metaphase plate.
But they're lining up as pairs, not as single chromosomes like in mitosis.
A critical difference. And the orientation of each pair is totally random. It's a coin flip whether the maternal or paternal chromosome faces north or south.
And we'll come back to why that randomness is so important. Next up, anaphase west.
The homologs separate. The maternal chromosome of the pair goes one way, the paternal goes the other. But, and this is another huge difference from mitosis, the sister chromatids are still attached to each other.
So you're pulling apart the pairs, but each chromosome you pull is still an X shape.
Exactly. Then you get telophase I and cytokinesis. The cell splits, and you now have two haploid cells. But because the chromosomes are still duplicated, we have to go again.
On to meiosis the second. And you said this part is much more like mitosis.
It is. Its only job is to separate those sister chromatids that are still stuck together.
So in metaphase summons, the individual duplicated chromosomes line up.
Right. And because of the crossing over that happened earlier, the two sister chromatids on a single chromosome are often not even identical anymore.
Then the final split in anaphase 2.
The proteins holding the sister chromatids together at the centromere finally break down, and they separate, moving to opposite poles. Each one is now considered its own full chromosome.
So what's the molecular secret here? What's controlling this two-step separation?
It comes down to a protein glue called cohesin. This stuff holds the sister chromatids together all the way from S phase.
Okay.
In anaphase 1, an enzyme comes in and cleaves the cohesin along the arms of the chromosomes. That lets the homologs separate. But the cohesin right at the centromere is protected. It stays intact.
Ah, so the sisters stay glued at the middle.
Until anaphase the second. That's when a different signal causes the centromeric cohesin to be cleaved, finally allowing the sister chromatids to separate. It's a beautifully timed two-step release of the glue.
And the end result of all this, after telophase 2,
4 daughter cells, each one is haploid, and each one is genetically unique from the parent cell and from each other.
we can even see this happen with data, right? The yeast experiment.
Yeah, it's a great way to visualize the reduction. Researchers measured the mass of DNA in yeast cells in femtograms. A cell in G1 starts with, say, 24 femtograms of DNA, let's call it X.
After S phase, it duplicates, so it's at 48 femtograms, or 2x.
Right. After meiosis A separates the homologs, the DNA content in each of the 2 new cells drops right back to 24 femtograms, or 1x.
Even though the chromosomes are still duplicated.
Correct. Then after meiosis the second separates the sister chromatids, the DNA content is halved again, down to 12 femtograms, or 0.5x. It's a perfect physical confirmation of the two-step reduction.
So all of this incredible molecular choreography, it has one massive evolutionary payoff.
The generation of huge amounts of genetic variation.
It starts with mutations, right? Those are the original source of new gene versions or alleles.
Mutations create the raw material. Sexual reproduction then shovels that material in 3 spectacular ways.
Okay, mechanism 1.
independent assortment of chromosomes. This goes back to that random coin flip in metaphase 1. The way your 23 pairs of chromosomes line up is totally independent for each pair.
So the orientation of pair 1 has no influence on the orientation of pair 2.
None at all. And the number of possible combinations you can get in a single gamete is 2 to the power's n, where n is the haploid number.
So for humans, n is 23, so that's 2 to the 23rd power.
2 to the 23rd, which is over 8.4 million.
Wait, 8.4 million possible combinations of chromosomes in a single sperm or egg, just from the way they line up.
Just from that, it's staggering.
And that's before we even talk about the second mechanism, crossing over.
Right. That creates what we call recombinant chromosomes, single chromosomes that are now a patchwork, a mosaic of maternal and paternal DNA.
Which means even the sister chromatids that separate in meiosis II aren't identical anymore. It reshuffles the genes on the same chromosome.
It adds a whole other layer of nearly infinite variation on top of those 8.4 million possibilities.
Okay, so that's 2. What's the third mechanism?
Random fertilization.
So you take any one of those 8.4 million possible sperm.
And you randomly fuse it with any one of the 8.4 million possible eggs.
And the resulting zygote is one of, what's the math on that?
It's about 70 trillion. 2 to the 23rd times 2 to the 23rd. And that's still not even counting the extra diversity from crossing over.
70 trillion. So when people say you're unique, it's not just a nice sentiment. It's a mathematical reality of an almost unbelievable scale.
It really is. And this diversity is life's insurance policy. Natural selection acts on it. A population with more variation is much more likely to have individuals who can survive when the environment changes.
But there's always an exception, isn't there? The weird case that tests the rule.
The veldoid rotifer, a tiny aquatic animal that seems to have given up sex entirely for something like 50 million years. They only reproduce asexually.
So how on earth do they get the variation they need to survive for that long?
They have a wild workaround. When their environment dries up, they go into a state of suspended animation, and their cell membranes can crack. When they rehydrate, their cells repair the cracks, but in the process, they suck in bits of foreign DNA from their surroundings.
So absorb it.
They absorb it and incorporate it into their own genome. They're getting genetic diversity by literally stealing genes from other organisms.
So even the great exception proves the rule. You absolutely need genetic diversity to succeed long term. Sexual reproduction just happens to be the most common and effective way to get it. So let's just quickly recap this amazing journey. We started with a diploid cell.
Duplicated its chromosomes and then ran it through meiosis I, the big reduction division, where the homologous pairs get separated.
Followed that with meiosis II, which is basically like mitosis, separating the sister chromatids.
And we ended up with 4 unique haploid gametes. The entire process, from the timing of the cohesin glue to the coin flip of independent assortment, is all designed for one purpose: to shuffle alleles and prepare the next generation for an uncertain future.
And that leaves us with a final thought, connecting all of this cellular biology to, of all things, the grocery store. The most common banana in the world, the Cavendish. It's triploid.
Meaning it has 3 sets of chromosomes, 3N, and for that reason it's sterile. It can only be reproduced by cloning.
So, for you listening, think about what we just discussed. If that precise pairing of homologous pairs is absolutely required for meiosis Earth to work, what does a third odd one out set of chromosomes do to that process?
It breaks it. The alignment can't happen properly, so you can't make balanced, viable gametes. And thinking bigger, how does that total lack of sexual reproduction, that complete absence of genetic variation, leave the entire global banana supply vulnerable to a single fungus? That's the evolutionary importance of meiosis, playing out right now on a global scale.